Hello everyone, it's nice to be back with you. We're going to continue with A3 and we're going to start with Lyme's disease because I told you that my sister got Lyme's disease and I do not want any of you to get Lyme's disease. And so let's get started on that. There will be some short answer questions about Lyme's disease on your second exam. So let's get started on Lyme's disease. Ticks. Has everybody seen a tick before? You know, there didn't used to be that many ticks around. Now there's a lot. I was just sitting in my front yard. I got a tick on my leg. I never saw a tick before I went to grad school. But the reason why ticks are so common now around here is because they cut down a lot of the oak trees. So why would that make a difference? Well, the reason why that makes a difference is because the ticks really prefer to suck on the blood of mice. And these mice like to eat acorns. And if you don't know, an acorn is the seed of an oak tree. So acorn is basically a baby oak. So we have a lot of oak trees. That means we have a lot of acorns. That means we have a lot of mice eating the acorns. And then the ticks, who prefer to drink the blood of mice, have a lot of mice to drink from. But come to our scenario now, cut down many oak trees. When I was growing up around here, there was hardly any buildings. There's many trees. There's farms around here. Most of what you see around here was not here like 40, 50 years ago. So they cut down the oak trees. Not as many acorns. Not as many mice. So the ticks have to go somewhere else to feed. And guess what? We're their hosts. So let's look at ticks. These are known vectors that transmit Lyme's disease. Vectors are organisms, doesn't have to be an animal, that are capable of transmitting diseases to humans. In the United States, Lyme's disease is actually caused by a bacteria. Alright, so it's not the ticks that actually cause the Lyme's disease, but it's caused by a bacteria, Borrelia burgdorferi, which is carried primarily by deer ticks. They're brown. Some of them are really tiny. They might actually look like a little mole or freckle. And it can be really hard to spot. And here's a picture over here of the bacteria. It's actually magnified 400 times. Lyme's disease is the most common tick-borne disease in the Northern Hemisphere. And here are some common vectors. Deer ticks are the most common around here. Now, here's some questions that are going to be on your short answer part. You need to know that the ticks feed on the blood. They attach to a host, and they feed until they're swollen. And I'll show you some pictures of them when they're swollen to their normal size. They're disgusting. They look like these disgusting gray raisins. Really gross. Now here's the important part. Only when they're feeding, and they're all swollen, only then do those ticks that carry the disease, that carry the bacteria, only then can they transmit the bacteria to the host. Or if the host is infected, then they can get it from the host as well. Now, many ticks do not actually have the bacteria. Depending on the location, anywhere from less than 1% to more than 90% of the ticks are infected with this particular bacteria. Spiral based on the shape. And here is a picture of a very, very swollen tick. So deer ticks typically feed on the blood of mice. That's their preference. Small birds and deer. But they can also feed on the blood of humans, cats, dogs, horses. They live in low bushes, tall grasses, and wooded areas, waiting for warm-blooded organisms to pass by, because they know they have good blood. Deer ticks are most active in the summer. So, you should know how you can get this. To contract Lyme disease, first of all, you must be bitten by an infected deer tick. The bacteria enter your skin through the bite, eventually make their way into your bloodstream. Before bacteria can be transmitted, a deer tick must take a blood meal. It makes you know this number, and that can take more than 48 hours of feeding, so 2 days. Only ticks that are attached to your skin and are feeding can actually transmit the bacteria. So sometimes you might have a tick that's attached, but it's not swollen. You're totally fine. An attached tick that has a swollen appearance may indicate that enough time has elapsed to transmit the bacteria if they have the bacteria. They might not. You want to remove the tick as soon as possible if it is attached. The infection is not transmitted from pet to pet, nor from pet to owner, only through an infected tick bite. Again, as I said, not all ticks are infected with this bacteria. Now you gotta be really careful of how you actually remove the tick. Let's say there is a tick that's attached to you and actually has the bacteria, but it has not yet sucked your blood. And you go out to it and you squeeze its belly. Kind of acts like a syringe, and you're going to be injecting that bacteria into yourself. So you got to be really restrained when you see a tick on you, especially if it's swollen. But if you have a tick and it's not swollen, again, be very careful. Don't squeeze it. Be very careful. But I'm not going to go through all of this. But here are some symptoms that you might have: it: fever, headache, fatigue. This is a characteristic skin rash. Looks kind of like a bullseye right here. But a lot of people don't get that. Most people don't. Right here it says to the left, characteristic bullseye-like rash forms at the site of the tick bite 3 to 32 days after being bit. That's as few as 9% of cases actually get that. But you can't take antibiotics. My sister who got Lyme disease, she had antibiotics right away, and there were no after effects of having it. And there is no vaccine available for this. Alright, I'm not going to go through all of this. I just wanted to show you some pictures, show you how small that could be. Deer ticks actually have a 2-year cycle. 2 years for one adult egg generation to mature. And they actually can be found all year long, even in wintertime. But you're usually going to see them more in summertime. Here it is. Different life stages. Adults. There's the nymph, larva. And they're going to feed once at each stage. And since the whole point of telling you this is to help prevent you from getting this, I am going to be asking you some ways to prevent yourself from getting this. So let's take a few looks at this. So ticks can be found in grassy or wooded areas, even in your own backyard. So here's the most important thing. Especially if you're going walking outside, especially nowadays when you can't go anywhere except maybe go walking Single most important thing you can do is check yourself for ticks once a day. Here's some favorite places to look. Between your toes, back of your knees, groin, armpits, back of the neck, hairline, behind the ears. Make sure you check your children, your pets, dogs, cats. Be especially vigilant after spending time in wooded or grassy areas. Again, some of these are so small, you might not see them. And help with a shower as soon as you come indoors if you can. That's often not an option. But remember, for 48 hours. And ticks are often going to remain on your skin for several hours before they actually attach. So probably like 3, 4, 5, 6 hours before one will actually attach if it's on you. Showering, using washcloth, combing your hair, may be enough to remove any unattached ticks. Another thing that if you can do, if this is convenient, as soon as you come in from outside, put your clothes in the dryer in high temperature for about 15 minutes. That's pretty successful. May through October is the time you need to get special attention looking for ticks. There's some other things you can decrease your risk of getting Lyme's disease. Protect yourself by wearing long sleeves, long pants, tucked into socks and boots. Well, it might be kind of hard thing to do in summertime. You're going to look awfully silly and you're going to be extremely hot. Light-colored clothing makes ticks more easily visible before it attaches itself. Stick to main pathways, center of trails when hiking. Attached ticks should be removed promptly. Not removing ticks promptly or properly can cause disease. Bacteria from a tick bite can enter your bloodstream only if the tick stays attached to your skin, again, 48 hours or longer. So if you remove a tick within 2 days, your risk of acquiring Lyme's disease is low. Now, if you do remove a tick, and it's not swollen, you actually might have a red mark on your arm, and it might itch for a while, maybe 1 or 2 weeks even. Does not mean you have Lyme's disease. So once you pull that tick off, you might want to clean that area. Apply antiseptic to it. They do have tick remover things. I know they have them at Walgreens. I'm not sure where else you can find them. But if you don't have any special tool, you can remove an attached tick with fine point tweezers. Grasp it right where it is when it's sticking into your skin and pull it right by its head. Pull it out straight. Don't squeeze it. Don't crush it. You can use insect repellents that are natural ones that work pretty well. Oil, lemon, eucalyptus, that works really well. And do your best to tick-proof your yard. Clear brush and leaf litter from the ground. Keep wood piles in sunny areas. Keep your grass cut short. If you have swing sets and patio furniture, keep them away from grassy areas or wooded areas. Swing sets, you might want to put them in sand, or you could even get, some outdoor carpet that looks like grass. And don't assume you're immune if you've already had it, because you can get it again. Alright, now we're going to start material for photosynthesis. First, we're going to look at the visible spectrum. And it is measured in nanometers. So, one way you can remember it is Rory G. Bibb, backwards. I can write that for you. That is an O. Rory. Blue. Indigo. Red, orange, yellow, green, blue, indigo, violet. So, visible spectrum, Roy G. Bibb, backwards. So if you look at this, here's the violet, blue, green, yellow, orange, red. And then if you look at the numbers, it goes from 400 to 700. Okay, so take a look at that. You will need to know the numbers. You will need to know the nanometers that go with the colors. And some of your photosynthesis lab experiments deal with this. So if you look at this larger spectrum, not just the visible spectrum, then that's our visible spectrum. Now this is a lot of different wavelengths, gamma rays, X-rays, UV. This little part right here, that's the only part that we can see. Infrared, radio waves, etc. So we can only see a very tiny bit of the spectrum. The spectrum are all the different wavelengths that are coming from the sun. And if you look at this top, right, these are the wavelengths. I'm going to draw you a wavelength here. So we get a different color. And I'll try that color. Alright, let's go to the middle of one, to the middle of another. Crest of one to the crest of another. That is our wavelength, and that's the symbol of it. That's a Greek lambda. And these are going to be in nanometers. I'm going to draw you another one. Okay, so if you see the one on the left right here, this is a larger wavelength. This is a smaller wavelength. As we have a larger wavelength, that's nanometers, we're going to decrease energy. It's hard to write like this. I think I'm improving though. Let's see that here. No, hold on. Right. So these ones right here, this over here, let me get a different color. And for just comparing these two, this has less, I'm just going to put a capital E for energy. And this one has more energy. That is an arrow going up. So these smaller ones have more energy. As they go up, bigger, less energy. Put the arrow here. As you go this way, you're increasing energy. This way, increasing wavelength. So light and chlorophyll. This actually is referring back to one of the experiments that you did as well in lab. So chlorophyll is green. Okay. You need to know why that's green. It's green because it's not using green. It's reflecting the green wavelengths. So it's reflecting these wavelengths. Look at my circle right here. It's reflecting these. It's not using those ones. Absorbs some wavelengths. So the ones that you do not see, those are the ones that are being used. So reflected means, right here. And if I write cursive, let me see. That's even better. Reflected means that's an R, not used. This means it is transmitted. I think I'll just print it. Transmitted. So it's not used. So the colors that you see, those are the ones that are not used. So the plant is green because it's not using the green. So let's look at this other word, absorbed. different color here. So absorbed. I've developed certain muscles to be able to write like this. This means used. This is important for your photosynthesis lab. Absorbed means used. Alright, used. These are not seen. These ones are seen. So for your quiz, lab quiz, used or absorbed. But when you look at a spectroscope, you do not see all the colors. So let's make it like this. R, Y, G, green, blue, B, indigo. Oh, that's I, violet. Let me just make little boxes here for each of them. And there's an I. So if you look through the spectroscope, so you have the spectroscope and you would first look through the spectroscope without any pigment in front of it, and you would actually be able to see all the different pigments. You'd see red, orange, yellow, green, blue, indigo, and violet. Then what you would do is you put a test tube with pigment in, and you put it right in front of the spectroscope, and you would not be able to see, let me get maybe black here. You would not be able to see the blue, the indigo, the violet. A lot of this would be gone. You would not be able to see that. And a little bit of the red probably would be gone. And maybe a little bit of the orange. So you would not be able to see this. The reason why you would not be able to see these colors is because these ones are used. You would be able to see those ones. So let's look over here, absorb some wavelengths, or it would be white. So white is when none of them are absorbed, so they're reflecting all of them. Black, two ways to get black. One could be that it's absorbing all the wavelengths, or it could be like an outer space and might look black if the particles are, very far apart and nothing comes and collides with them. Alright, so from this picture, right there, these ones here have more energy, they're shorter wavelength. These ones on the red side, longer wavelengths, less energy. Chlorophyll is green because it's reflecting green. It's not using the green, it's transmitting the green. It's not the other colors because those colors are being absorbed and used. So what we're going to do right now is we're just going to go through a short summary of what takes place in photosynthesis, and then we're going to go into more detail, step by step. Find a picture like this on the internet, change it a little bit to how I like it. Let's look at this. So photosynthesis, first of all you need to know what is photosynthesis. Anybody remember what photosynthesis is? Just looking at the word synthesis, photo. Remember the plants are the autotrophs? So they're synthesizing their own food using light. Remember the autotrophs are able to convert inorganic molecules to organic molecules. So let's look at this. You need to know what goes in and what goes out. So let's say in, I guess I can use it. Here's in. And I'll just use the same color to make it easier. Out. So if you look at the formula, we know carbon dioxide and water, sunlight, this green arrow right here, there's some chlorophyll right there, and that's going to be converting it into sugar, and we know that sugar is actually glucose, right? So we know that solar energy, come on, solar, it is an R, energy, and look over here, we know that carbon dioxide is going to go in. Remember that carbon of the carbon dioxide becomes a carbon in the glucose. Water is going to come in from the roots. And what's going to come out is oxygen and glucose, C6H12O6. So let's take a look at it. Here's the sun, solar energy. Alright, solar energy, so here it is. Solar energy is going to be absorbed by the chlorophyll inside the chloroplasts. And those are going to be in the leaves, wherever it's green. Carbon dioxide is a gas that's going to go in, right, into the leaves, through the little pores, the stomatas. What's going to come out is the glucose, the sugar. Remember, that's chemical bond energy, right? That's going to get stored in the plant. It's carbohydrates. And also, it's going to be releasing oxygen to the atmosphere. And then here's us. There's a nose. I had to make a big nose. That's why I made a big nose like that. You can know it was a nose. So we're going to breathe in that oxygen. And it can go around and around. Oxygen, carbon dioxide. There's the water coming in from the roots. Remember transpiration from test 1? So the sun is going to suck up the water, kind of like someone's sucking something up from a straw. Alright, here's a little bit of the history of photosynthesis. Here's our formula. 1772, Priestley discovered that oxygen is produced. 1779, they found out that sunlight is needed. 1782, they found out carbon dioxide is also necessary. Then they found out water is an ingredient. And then they found out that sugar is being produced. That's how they came up with the formula. So it actually took quite some time to actually come up with this formula. 6. Let's do 6, 6, look, 6, 12, 6, 6, 6. So that should be pretty easy to memorize. 6 carbon dioxides. Notice there's 6 carbons. The carbon dioxide over here. 6 carbons and glucose. These carbons of the glucose come from the carbon of the carbon dioxide. So let's look at some pigments that are involved. Chlorophyll we know is green. And you do need to know the numbers. Reflects green wavelengths. 500 nanometers in width. Absorbs the other wavelengths. So the ones that you see are the ones that are not being used. We actually have different kinds of chlorophyll. And they're going to be absorbing different wavelengths. Chlorophyll A is dark green. Chlorophyll B light green. Chlorophyll C blue green. Chlorophyll D yellow green. Now if you went outside, especially in summertime, and you'd see all these different plants and they're going to be green. But a lot of them actually are not going to be the same color green. And the reason they're not going to be the same color green is because they're going to have different amounts of these different kinds of chlorophylls. That's kind of neat. Also from the photosynthesis lab, you would use something called a spectrophotometer. And in the spectrophotometer, you would come up with some numbers based on absorption and transmission. And we'd have two absorption peaks. Those are the ones they're using it the most. And yours would look kind of like this. Chlorophyll A, right here. It would look kind of like these two right here. Notice the colors, blue, violet, indigo, and red right here. So, you might want to study these numbers right here and the two absorption peaks. Alright, I skipped a little bit. Now we're going to look at autumn leaf colors. Why do leaves change color in the fall? Well, here's the ongoing idea, is that chlorophyll is the predominant pigment in most plants. So all the other pigments are there, but you can't see them because of the dominant chlorophyll. The idea is also, though, that the chlorophyll is most sensitive to the decrease of light intensity in the fall. So the idea is that the chlorophyll pigments will die before the other pigments. So once they die, then you're able to see the other pigments that were there already the entire time. So I'm not going to go through this. Down here, this picture, you can look at that on your own. So let's continue on after that. Okay, so let's look at the plant leaf. Plant leaf is an extremely important part of the plant. And if I ask you, what is the function of the leaf? You might think about it, but you probably say to trap energy from the sun. It's probably what you would say. And this is a picture of a plant. Does anybody know what kind of leaf this is? What kind of leaf is that? What's this brown thing here? I mentioned them earlier when we talked about ticks. This is a type of oak leaf, and these are acorns. And that's the seed of an oak tree. So again, they're designed to trap energy from the sun. Here's the blade, petiole. So not just grass is called blade, like a blade of grass, but this is also called a blade. So this is an important part for you to know. You'll need to know the different parts of this leaf, and you'll need to know this for your lab exam as well. So leaf section, main organisms of photosynthesis. Alright, upper epidermis and there's a lower epidermis. These are just the single cells, layers, the top and the bottom. And I'm on B right now. They're often covered with a waxy protective material to decrease water loss and injury by invaders. So that's waxy covering. That's called a cuticle. And the inside, you can really see it on like really shiny plants. Like really shiny plants, they're going to be having a thick cuticle. So that word right there, cuticle. Meso means middle. Alright, so here we have the two outside, kind of like peanut butter sandwich. We got our bread. Those are like the upper and lower epidermis. Then all this stuff inside here, this is whatever you might want to put on your sandwich. Peanut butter, jelly, raisins. I don't know, we used to put pretzels in there when we were little. So, meso means middle, so the stuff that's in the middle. There's two main layers of the middle. So we have a palisade layer. Palisade, they're called palisade because it looked like guards standing up straight. Look pretty crowded, right next to each other. And this is where most of the photosynthesis is going to be taking place. And that is because that is where most of the chloroplasts are found. If you look over here, actually more than 80% of the chloroplasts of the leaf are actually found right here. Those are little dots. You know, they should be green, but I didn't have green at the time. The green ran out. Spongy layer. See, I made some spongy looking things. If you look online, you can see a lot of pictures of this. Now make sure you know all the different functions of the spongy layer. Storage of photosynthetic products. What are the products? What were the things that came out? That was the glucose and the oxygen. Gas exchange. What gases are involved? Look at the formula. So think about it. Tell me, what gases are going to be involved here? What gases went in? Carbon dioxide. Water, maybe. Water gas. Oxygen. Alright, so gas exchange. And these have some chloroplasts. Should be a few dots right there. So they do have some photosynthesis. So storage of the glucose, gas exchange, carbon dioxide, oxygen, and some photosynthesis. And here's the lower epidermis. Now look over here, some words here. Here's one word that I warned you about, stomatus or stoma. Remember, chloroplast, stroma, stoma, right, is right here. So let me draw you a picture. I'm going to draw a picture of your mouth. Alright, here's a nose, here's some lips. That's the top lip. Not too bad. There's a nose. That's a nose. Eyes. These are eyes. Okay, so if you think of the stoma and the guard cell like lips, right? The guard cell like your lips, right? So these are your guard cells. Okay, now I'm going to try to open my mouth here. Erase. Oh, I erased all of it. Didn't mean to erase everything. Ugh. Alright, well, let's start over here. Alright, so here are my lips. This is closed. These are your guard cells. You want to open up your mouth. Alright, pretend here's your lips. I'll do it this way. You open your mouth really wide. Okay? That hole that you make, go ahead and do that right now. These are your lips. Here's your, so you open your mouth, and that hole, when you open your mouth, that's the stoma. Alright, so the guard cells are like your lips. So close your mouth. Can't take it, no breath is coming in and out. Nothing's coming out. Open your mouth. So you can go in and out, in and out. So the guard cells are what control what goes in and out. Alright, so they have humidity control, or if there's a lot of pollution, they'll close. If it's really, really hot out, they're going to close up. Everybody understand? I just moved it down a little bit so we can see the last part of it. So let's go back to the spongy layer. We said gas exchange, so we have our formula. Carbon dioxide is a gas. This could be water vapor form. Not necessarily. More apt not to be, but it could be, like when it evaporates. And oxygen. So those can all be involved with this gas exchange. Storage of photosynthetic products. There you go. Okay, let's look at veins. And here's a picture. I guess I could draw a picture of it. If you ever went to a tree nearby and you took off a leaf and you like stripped it and it had all these like, why is this going on? And all these things sticking out, and they might have them like that. Okay, those are the veins. Well, here's a really good experiment for you to do, especially if you have children. You can get some celery, or I guess it doesn't have to be celery. The celery works pretty well. Just try what you have. And put it in something that is colored. If you have food coloring or something like that. And you might want to take different colors, a few glasses of different colors, and stick it in there and wait a few days and see what happens. Check on it every once in a while, see what's happening. And you'll be able to see where things are being carried. So the leaf, right? Very, 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 very important. Again, review of what a chloroplast looks like, because you're going to really need to know the different parts of this chloroplast to understand what's happening. One chloroplast contains all the enzymes that it needs for photosynthesis to take place. It's all right there in the chloroplast. Chlorophyll within the thylakoids, remember each of those little green pancake things, that's where the solar energy is going to be absorbed, and that solar energy is going to be energizing an electron. So the energy that it traps from the sun goes into an electron. Now here's our word stroma. Alright, here's our chloroplast. That's out here, outside the thylakoid. That area is called the stroma. And out here, right out here is where the carbon dioxide is going to be changed into glucose. It's going to get reduced and changed into glucose. So thylakoids, this is where we're going to have something called the light reaction. The light reaction is the reaction that needs light and actually traps the light, solar energy. Stroma, what we call the dark reaction, the Calvin cycle, is going to take place out here, and this is where the carbon dioxide is going to get reduced and converted into glucose. Remember, granum is a whole stack of these thylakoids. Since we have 1, 2, 3, 4, we can say we have 4 grana, Latin. So now we're going to actually be using some of that chemistry that we learned for test number 1. And we're going to review oxidation and reduction. These are going to be extremely important in our energy conversions, both in photosynthesis and in cellular respiration. So let's look here. A, we know that photosynthesis is converting solar energy or light energy into chemical bond energy. It is converting carbon dioxide into glucose, C6H12O6. It's going to start out with these two inorganic molecules, carbon dioxide and water. Remember, there's one way to abbreviate glucose, and it's making glucose. Inorganic, inorganic, and organic, and then inorganic. And the way this is going to work is through oxidation reduction reactions. And we're actually going to be adding on to what we learned for test number 1. Reduction. We were just referring to electrons earlier, but now look, reduction is a gain of 3 things. Make sure you know all 3. Electrons, hydrogens, and energy. So reduction, gain of 3 things, electrons, hydrogen, and energy. Oxidation, we just learned it as loss or removal of electrons, but now we're going to say it's loss of electrons, hydrogens, and energy. So A, let's say as an electron, it's not just going to throw it out there, it's only going to throw it out there, give it away if there is someone to receive it. So there it is. So if you look at my little box here that I made for you, reduction, gain, oxidation, loss. Reduction, gain of three things: electrons, hydrogens, and energy. Oxidation, loss of three things: loss of electrons, hydrogen, and energy. So let's go to step number 4. Synthesis of glucose from carbon dioxide is through the reduction of carbon dioxide. So if we go back up to our little box up here, let's do a different color here. So we said it's reduction. Oh, that is not very straight at all. I don't know if I can go Control Z. Reduction. So let's go. Oh, that little box is in the way. I'll get rid of it. So this is reduction. So look at carbon dioxide. That means carbon dioxide has to receive those three things: electrons, hydrogens, and energy. So carbon dioxide is going to get reduced. Carbon dioxide is going to receive electrons. It's going to receive hydrogens. It's going to receive energy. And it's going to be formed into glucose. Okay, let's take a look at water. Water is oxidized in photosynthesis. Water, oxidized, loss of electrons, hydrogens, and energy. Look, there we go. Hydrogen, oxygen, and 2 electrons. Energy released from the redox reactions powers the formation of ATP. So oxidation reduction in photosynthesis, also in the formation of ATP in cellular respiration. And now we're going to look at 3 types of phosphorylation, or 3 ways to make ATP. Substrate phosphorylation. So let's review this. Straight line. Remember, we had a wavy line. Wavy line because the phosphates are negative. It takes a lot of energy to hold them together. Alright, let's say we want to make ATP using substrate phosphorylation. Alright, so then what we would have is we would have a molecule. It's just a molecule, could be anything. And this molecule has a phosphate on it. Okay? So substrate phosphorylation, here's the substrate right here, a molecule. We're going to take it off there. That phosphate's going to go here. And now we're going to have ATP. So substrate phosphorylation, take the phosphate off some kind of molecule here, substrate, put it onto ADP, now we're going to get ATP. Oxidative phosphorylation, that is going to be dealing with the electrons, electron transfers, redox reactions, and we're going to have something called an electron transport system, or you might see it as electron transport chain. So for us, that's going to be in our electron transport system. Electron transfers. So, let's take this picture of a staircase. And if you're talking about respiration, the electron come from water. If we're talking about non-cyclic reaction in photosynthesis, so non-cyclic light reaction of photosynthesis, we get our electron from water. So here it is. If anybody ever played hot potato, okay, normally we actually would do this in class. First, let me get rid of this, and then we'll play hot potato. Alright, I'm gonna make some stick creatures here. Okay, this is you, students. Alright, this is you. Come on. Oh, I didn't mean to look like this. It doesn't do what I say. I guess I should practice with this. I'm just trying to make stick creatures here. Nothing personal if it doesn't look like you. Alright, let's review. Come on. Oxidation. Reduction. Oxidation. Loss. Gives it away. It's an S. This is a recipient. Alright, recipient. Wish they just let me go backward one. That is a P. Well, we're just going to leave it like that. Okay, so let's say I have a marker. And I gave a marker to this student right here. Alright, so here's me. Okay, there I am. I gave the marker to this student. I was oxidized. This student was reduced. Then this student gave to this one. So now this student is oxidized. This one is reduced. That student gives it to this one. This one now is oxidized, and this one is reduced. This one here is reduced, and I give it away. Now that's oxidized. When they receive it is reduced. Now this one was originally reduced, now it's oxidized when it gives it away, etc. So and every time gives it away, some energy is lost. Okay? That's pretty much what the electron transport system works like. Electron passed on, kind of like hot potato. Every time, energy is going to get released, and it's just the perfect amount of energy that the cell needs. If it was too much energy, it wouldn't work. Kind of like my twin sister and I were little, she put a bobby pin in the electric socket. Hair stood up a little bit, and her fingers got burned. It's too much energy. And right here, electron transport system, right? Whole series of oxidation, reduction reactions, and this is going to be used to make ATP. Let's go to the next picture. I skipped a little bit because if you understood the picture, right, those words were just a description of the picture. So if the energy was released in one big step, most of it would be lost as non-usable heat. And now we have some new things right here. NADP plus and NAD plus. We're going to call those electron carriers. Alright, electron carriers. Electron carriers of reduction, oxidation. And photosynthesis is going to have some. And so is cellular respiration. The one that has the P, NADP plus, that's the one with photosynthesis. So that's the easy way to remember it. NADPH. Alright. Notice here, now it's NAD plus, that means it has to receive it. NADPH, that means it already received whatever it's supposed to receive. Remember those three things: electrons, hydrogens, and energy. NADPH, it's carrying it, it's like a taxi, carrying it. It's going to give away a hydrogen, electrons, and release some energy. So donates the hydrogens, electrons to a substrate, so it's going to get oxidized. Remember, oxidized, give away. Chloroplasts, electrons that enter the electron transport system, are taken from water. This is in the non-cyclic. One reason why you need to water your plants is because you need the electrons. Those electrons that come from water are going to be energized by solar energy. So solar energy is going to go into the electron of the water. Following electron transport, NADP plus accepts electrons, hydrogens become NADPH. So it's kind of like a taxi. Pick it up, drop it off. Pick it up, drop it off. Pick it up, drop it off. NADPH supplies those three things that we need: electrons, hydrogens, and energy. And that is going to be needed, right, to reduce the carbon dioxide to the glucose. So here to pretend these are the students, a potato. In this case, instead of me, it's NADPH. And now here's another one, FADH2, in cellular respiration. This is also called the cytochrome system because each of these molecules is called a cytochrome. Let me spell that for you. Let's see. CYTOCHR. I must be doing something wrong in making this. Cytochrome, here's an R. Cytochrome system. Okay, now we're going to talk about an extremely important enzyme. And if you look at it, ATP synthase, you can probably figure out what it's for, right? Synthase, synthesis, synthesize. So it is the enzyme used to synthesize ATP. And we know the only molecule that we use for energy is ATP. So without it, no life, right? And let's go down to the description. There's a few pieces of this description that you will need to know. And I'll point out the ones that you actually need to know. Every cell contains hundreds of miniature motors embedded in the surface of the thylakoid and mitochondria. So electron transfer system is in the thylakoid, is in photosynthesis and in cellular respiration. So each motor is 200,000 times smaller than a pinhead, extremely small. The motor, okay, make sure you know what happens here. Inside it, there's a motor. So you need to know there's a motor in there. Forward, you can just say it makes the bond between ADP and P to form ATP. And know about this little wheel. At the center of ATP synthase is a tiny wheel. You don't need to know how many times it turns around. But at the center, there's a little wheel. That spins around. 100 revolutions per second. That's really fast. And you need to know every time it spins around, it makes 3 ATPs. So ATP makes a bond between ADP and P to form ATP. At the center of ATP is a tiny wheel, and every time it spins around, it makes 3 ATPs. Each enzyme is made of 31 separate proteins, made of thousands of precisely arranged amino acids. This is an amazing molecule. Just one mutation, one change in any amino acid, in any part of this molecule, and the motor is useless. You can't make any ATP, and you can't have any life. So this is very extremely important enzyme. It's absolutely amazing. Okay, now we're going to be looking at something called chemiosmosis, right here. Chemiosmosis. So you have to remember what the chloroplast looks like. Okay? Here's the thylakoid space. This is inside a thylakoid. Here's the thylakoid membrane. Here's the stroma. Everybody remember what it looks like? On the thylakoid membrane, we're going to have 2 locations that are going to be trapping energy from the sun. We're going to call them photosystem 2 and photosystem 1. So they're going to be trapping energy from the sun, and they're going to put the solar energy into the electron. This actually is a picture of something called non-cyclic light reaction. In this case, the electron is going to come from the water, as I mentioned earlier. The electron is going to go into photosystem 2, where it's going to get out so much energy it can't stay where it is. It's going to deliver it to the electron transport system. Remember, the electrons go down the electron transport system. And as they go down the electron transport system, remember a series of oxidation and reduction. Every time they get passed on, they're going to release energy. So look at my note up here. As electrons are passed down the electron transport system, the energy is released. And that's going to be used to pump hydrogens from the stroma out here into the thylakoid space. And they're going to be building up. So right here, energy from electrons, as they are passed down the electron transport system, are used to pump hydrogens inside the thylakoid space. Okay? Let's go to the bottom picture. And this will be the last thing for today. Okay, let's start out with some water. Water is oxidized. Gives off an electron. We're just going to follow the electron. And here's the hydrogen. See, the hydrogens are building up here. Alright, so here, this electron actually is going to go here. Photosystem 2, PS2, photosystem 2. We're just going to follow this first part. We're not going to follow the second part. So electron goes to photosystem 2, where the solar energy is going to be put into that electron. Now that electron has a lot of energy. It's going to get passed down the electron transport system. As it gets passed down the electron transport system, series of reactions, energy is going to get released. That energy is going to be used to pump these hydrogens on the inside. So notice, all these hydrogens are positive. And we know positives, same, right? Positive, they're not going to want to be by each other. Right? They're going to try to avoid each other. Also, they want to have equilibrium. They want to have equal amounts on both sides. But they keep getting pumped inside. Active transport causes a high concentration of hydrogens on the inside. And there's only one place that the hydrogens can return to the stroma in their attempt to reach equilibrium. Building up, building up, building up. Oh, we don't like this. We don't like this. Where can we escape? Where can we escape? Ah, here, we found an escape. ATP synthase. So like, hey, we found a way. Let's escape. Go through here. Now remember there's motors inside here. The motion of the hydrogens going through. That motion of the hydrogens going through provides the energy for the motors to spin around. As the motors spin around, they make a bond between ADP and P to make ATP. That hydrogen might get picked up by NADP+, but it might get forced back inside. So these hydrogens might be going around and around. So let's start over one more time here. Let's review this. This whole process is called chemiosmosis. So look here. This is the non-cyclic light reaction. I know that because we're starting out with water. Photosystem 2. So water is going to get oxidized. The electron is going to go to photosystem 2. The solar energy is going to be transferred to that electron by all the different pigments in photosystem 2. So again, all the pigments in photosystem 2, they're going to be trapping energy from the sun, and they're going to send it over to that electron that came from the water. So that electron is super energized with solar energy. It gets taken over to the electron transport system. As it goes down the electron transport system, remember the hot potato, it releases energy. What is that energy used for? The energy is used to pump hydrogens inside. So let's say there's hydrogens here, it gets pumped inside. Hydrogens are building up. They don't like it. What are 2 reasons why they don't like it? One, they're all positive, right? Same charge. Like 2 magnets, put 2 positive sides together, they're going to be what? Repelling each other. And also they want to reach equilibrium. So they're trying to get away from each other. There's only one place they can make a break for it. And that is ATP synthase. And we know inside ATP synthase there's a little motor there. Hydrogens, in an attempt to escape, go through the only place they have a way to escape. And as they go through, that motion provides energy for that motor inside ATP synthase to spin around. And as that motor spins around, it makes a bond between ADP and P to form ATP. And we will review this next class. So next class we'll be starting out with B3 and I will review this. Alright, well have a great weekend and I'll post you some questions about this. Alright, until next time, be well.